Hi, welcome. My name is Francisco Villalobos and my professor Carlos Francisco Mendez from the Faculty of Science and Center of Genomics Science. I'm going to be talking about natural language processing for microbial regulatory interaction extraction. For this, I'm going to introduce key concepts, that is NLP and text mining, transcriptional regulatory networks, RegulonDB, and TESS. The objective of our work was extending tests for curation of regulatory interaction from biomedical text, mainly E. coli, to help maintaining RegulonDB using BioNLP. So what is NLP? It stands for Natural Language Processing and refers to a combination of statistical, machine learning, and deep learning models to enable computers to process human language. Well-known examples, it's Google Assistant, Alexa, Siri, the autocorrect in text messages, sentiment analysis, for example, in Twitter posts. And in our case, we were interested in biomedical event annotation, that is to process sentences from the literature. For example, here we can see that protein A is the cause of the binding of protein B and C. A lot of models have been built in this regard, and there are some competitions that are called shared task. And here we can see the topics of these models. For example, epigenetics or drug interactions. But what we were interested is in transcriptional factors. So transcription factors provide a rapid response to external threats, allowing the bacteria to survive. Here we can see an example, MAR-A is the multiple antibiotic resistance. And we can see that it blocks the expression. So the data we have, for example, that MAR-A is in this sentence, and the extracted interaction will be just to keep the information. And we have curated text from E. coli and Salmonella difficurium. If we collect all that interactions, we could build a transcriptional regulatory network. And that is what RegulonDB has done for more than 20 years. And here the TESS is the software that is published as a freely available open source from the Turku University. And it uses NLP. It's written mostly in Python, and it has performed well in these competitions to build the models. Here's an example of a graph output from TESS. The sentence is processed, and we could see that it has nodes and edges. The nodes are the trigger words, and the edges we can see, for example, here that it has a theme and a cause. So the research question is if we could adapt TESS using one of the models in the shared task to extract regulatory interactions. And the answer is yes. We could see here, for example, a very simple case in which the graph given by TESS can give the binary relation in the transcription factor and the gene. Just follow the cos edge backwards, and we could arrive. Here's another example, more complicated, but not too much, just the same. We took the blue edge and follow it backwards. So the algorithm, in essence, is just to keep track of these elements that have the type cos. And the sync nodes are the nodes that no longer have edges pouring through them. So the algorithm is to start from the nodes that has the cos and try to arrive at sync nodes. And the results, we could see here that, for example, more complicated graphs and sentence. Here, for example, is reduced expressions. So the algorithm here, for example, finds these three regulatory interactions. With comparing to our dataset, we see that we have more than 50% in true positives and in recall. And some perspectives, it could be improved. We did improve the scores by cleaning the reference data. The model was not built for the transcription factors. We use a lot of synonyms. For example, CRP complex, just CRP. And other special cases arose, for example, articles that used mutants, so in the expression. And these are the references, and thank you for your time.